Cryo-EM, or Cryo-Electron Microscopy, and X-ray crystallography, these are two of the main ways that scientists get a look at what proteins and various complexes look like. And when we talk about getting a look, it's not as easy as just going and looking with your eyes or even with a normal light microscope. That's why we have to use these really fancy techniques, is because the things that we're looking at are really small. And so crystallography and cryo-EM take different approaches to try to solve this problem of how do you get a look at these things, where the things that you're trying to get a look at is like the insides of proteins, which are made up of atoms, which are really close together. They're like thousands of times closer together than the wavelength of visible light. And so visible light is not going to help you visualize it. And so instead you have to choose more energetic sources. And so this would be an X-ray and X-ray crystallography, or electron beam waves in cryo-electron microscopy. And in both of these methods, you're going to need a lot of molecules, because you're going to need to kind of have signal coming from lots and lots of these different copies of the protein or of the complex in order to have enough signal to, well, see anything. And so in X-ray crystallography, you actually have all those copies are lined up in the same orientation, over space, in what we call a crystal. We'll get more into what this means. And then what happens is that the signal from the X-rays, when you beam those X-rays at it, they're going to get diffracted by the atoms in those molecules. And we'll talk more about what this means, but basically they scatter those. And then those scattered X-rays, they're going to interact with one another. And when they interact with one another, they can either build up constructive interference or cancel each other out, which you call destructive interference. And it's kind of like if you were to drop two golf balls into the water, and then you see the ripples, and then there's places where the ripples look bigger, and then there's places where there's anti-ripples. I don't know, what do you call it? But anyway, the waves are gonna add together. They're going to diffract in specific locations, and then if you have a detector, it can detect those strengthened X-rays on this detector, and you get this pattern of spots called a diffraction pattern. And it turns out, using a lot of complicated math, you can actually work backwards from that diffraction pattern to figure out where all those atoms were that were sending the signal. And you're able to do this because of the crystal form of the protein, that you have all those copies in the same orientation, so they kind of make these virtual planes and all this confusing stuff. But bottom line is, having them all in that same orientation is really important, but you only get them in that same orientation, and often proteins don't really want to sit still like you want them to in a specific orientation. And even if they do, that might not be the actually most biologically relevant orientation. Proteins, they like to move around. And so another technique, cryo-EM, would say, okay, you move how you want to move, and we'll just freeze you in place however you are. And so cryo-EM, it's more like you're taking actual pictures, but they're not really pictures like you would take with a camera. They're what we call projection images and stuff. But basically, you get these things called micrographs, where they look like really blurry pictures. And so you have all these molecules. What you do is you freeze them in this really thin layer of ice, this vitreous ice. So it's basically like glass, but it's water. And your molecules are oriented in all different ways. And then you send the electron beam at it. What's going to happen is that when you get those images, basically the molecules are going to be in all their different orientations, and then you kind of see something kind of like a shadow from where it was. And so depending on the orientation of the molecule, you're going to get different looking images. And so then it takes a lot of computational power to try to say, okay, well, which of these molecules look alike? Well, first of all, which is this is just crud, and what if this is actually your protein or your complex of interest? And then what you have to do is you have to the computer, so you pick those particles, we call them particles, these individual, the molecules or whatever you're looking at. That's why we call it single particle cryo-EM, as opposed to tomography and stuff, where you actually look at slices. But anyway, What you're looking at here is kind of like taking slices out of a protein, like in a CT scan, because you're getting all these different orientations. And then the computer has to kind of figure out, okay, well, which of these, if I rotate it this way, is this the same as that? And because these proteins, not only can they be in like different orientations, like standing up or laying down, but they can actually be in different conformations, so different shapes. And so remember, proteins like to move around, they're really dynamic, and so they can have different shapes or conformations. And so that's another classification that you can do, is you can kind of tease out, okay, well, which of the ones are doing a handstand and which of the ones are like laying out straight, or I guess those would be kind of similar, but which of them are doing like a star versus an L or something like that. So in this way, you're able to see multiple conformations. The problem is that you need to have enough of each of them in those conformation in order to have enough signal, because what you're going to be doing is you're going to be averaging the signal from all of those different molecules. And so by averaging all of their signal, you're able to get a strong enough signal that you can actually make out the atomic positions in that molecule. And so in both of these techniques, you're relying on lots and lots of copies of the exact same thing in order to get the information about what one copy of it looks like. And depending on the quality of the data, it's going to be easier or it's going to be harder, because what you get is you don't directly just get like, oh, this atom's here, this atom's here, this atom's here. Instead, what you get is you get a map. So in crystallography, what you get is like an electron density map. And in Cryo-EM, what you're getting is like a Coulomb potential map. Basically, the X-rays or the electrons are going to be interacting with kind of like different parts of those atoms. And so when we talk about crystallography, the diffraction, so like the actual like scattering from those different atoms, it's coming from the electrons. So we're not shooting electrons at them, but the electrons of those atoms are interacting with the X-rays we're shooting at them. And this is going to like deflect the X-rays that then can add together. When we get the diffraction, we get those spots. And then when we work backwards, what we get is the location of the electrons, because the electrons were what were scattering those X-rays, what were interacting with those X-rays. And so we get an electron density map. With Cryo-EM, what we get is a Coulomb potential map. So basically the electrons, they're interacting not just with the electrons in the atoms, but they're also interacting with like the nucleus of the atom. So like the positive charge, because what you're getting is the electrons that you're shooting at them are going to be interacting based on like charge and stuff. Complicated thing. In both of these cases, what you're ending up with is something that looks like a sausage. And if you have a high resolution data, you have a crisper sausage, and if you have a low resolution data, you have like a fatter sausage. And these sausagey maps, this is what scientists actually build the atomic positions into. You build those like stick figures and those ribbons and stuff. All of that is built, those are models that are built into the maps. And so the crisper that map is, the higher the resolution of that map, the better chance you have of sticking the atoms into the right places in that model. And so resolution is going to refer to how close together two things can be. Well, you can still like tell they're separate things. So the lower number is going to be a higher resolution, because this is basically saying like, I can tell that these are apart at a high resolution. So a low number, I can say like, okay, these are a centimeter apart. But what if you only have a resolution of like a meter? Then you wouldn't be able to tell that those were two separate things. What we're dealing with here is typically angstroms, and an angstrom is 0.1 nanometers, and a nanometer is going to be a trillionth of a meter. So we're talking really tiny values, really tiny distances, and so we need that really fine resolution. And so the lower the number you see, the higher the resolution, the crisper that map is, the easier it is to place those atomic positions in there accurately. Now, of course, you then have to actually go and place those atomic positions in there. And so there's software that can be used in order to do this, but then the scientists really have to go through like atom by atom and try to make sure that everything is actually like fitting nicely. And this is especially an issue with X-ray crystallography. Is what happens is that the map that you get is missing key information, or the initial things that you get, the information you get is missing key information. Because what that diffraction pattern represents is that's going to be the signal from a kind of mega wave that happens when those X-rays diffract. So when those X-rays get scattered and then they interact with one another, they're going to hit a detector. And when they hit that detector, you get the information about the intensity, so how strong that was. But you lose what's called the phasing. So just like you could have a wave whee, or you can have a wave hitting like this. Basically what happens is you don't have the phasing. You don't know if it's in step, if those waves are what part of the step they're on, type of thing. Hopefully the image makes it clearer. But basically, to deconstruct that mega wave from that pattern of spots, you need to know the phasing as well as the intensity, and you're missing the phasing. So what you have to do is you kind of have to say, okay, well, if the structure was like this, what would the phasing be like, and does this match? And so with crystallography, your map is actually going to change as you build that model in, because you're going to improve your model, which is going to improve the map, hopefully, and then back and forth and back and forth. Because what you're doing is you're basically saying, okay, you're trying to figure out what the map should look like if your model is right. And then if it's not right, then the map will look worse, and then you change it and that sort of thing. But the bottom line is you have experimental, you have to actually have some modeling built in when you're making that map. So yeah, it can get really confusing with crystallography maps. It's a little more straightforward with the Cryo EM maps, but neither of these techniques are just easy peasy. When you have a normal light microscope, there's all those glass lenses that are actually focusing the beams after they go through the sample, then you have to focus it to actually get those beams back and see the image and stuff, all this weird optics. But basically, you can't do that focusing with X-rays. And so this is why you have to rely on those dots themselves rather than having lenses then focus those back, and so you can see what you started with. And so you have to work backwards from those spots in order to figure out what you started with. With the Cryo EM, with electron microscopy, here you can focus the beams, but you can't do it with just glass lenses like you would with visible light. Instead, these machines use these series of magnets to focus those electron beams. So you're using these magnetic lenses rather than glass lenses. But then you're able to get these actual images rather than just a pattern of spots. But these images, their signal is going to be really, really faint, and you're going to have to get a lot of images in order to get a structure and things like this. But yeah, so that's one of the differences between X-ray crystallography and Cryo EM, and why you're getting these spots for the X-rays, but you're getting these blurry pictures for the Cryo EM. And so with these techniques, there's been recently a big upsurge in Cryo EM. This was because of some technical innovations, both in terms of first the cryo part is being able to do this really cold, so you're able to protect these samples because you're going to be shooting electrons at them, which are really energetic, and so this can be really damaging to them. And so one of the biggest problems, the biggest hurdles to trying to get good resolution is that you're frying your samples, basically. So by cryoprotecting them, it's able to help out. The other is being able to detect those electrons. And so there's these direct detection of electrons. That was a huge game changer, trying to be able to detect the electrons as opposed to having to have them on film that then you had to process or whatever. This is direct detection. It can actually detect the hitting of those electrons after they go through the sample. So cool stuff, and really helpful. Also really helpful was phasing, phase plates and things like this. Basically what happens is with Cryo EM, one of your biggest problems is contrast, phase contrast and stuff. Basically the molecules kind of blend in with the background, and it's hard to tell what is sample and what is what's your particle and what's the background. And so technique, the phasing and phase plates and things like this have really made it so that this is easier to do and more possible to do at all. But you still have that contrast issue. So with the X-ray crystallography, your biggest issue is trying to figure out that phasing, and with cryo-EM, your biggest issue is also often trying to figure out like the contrast. So what is my sample? What is the particle versus the background, because they kind of blend in together. And also just various software, lots and lots of technological improvements on the software side for cryo-EM, as well as there's been a lot of improvements in X-ray crystallography. But yeah, so cryo-EM, basically a few years ago, you could only use it for like really, really big things where it's a lot easier to tell that this is not background, this is actually my thing. But now it's with the advancements, you're seeing it go down and down and down in the size of the molecules that you can actually image, as well as seeing the resolution of those structures improve. So cryo-EM, it's often used to solve the structures of complexes, or like really big stuff. Whereas with X-ray crystallography, it's harder to get the structures of complexes, because those complexes, well, if one protein doesn't like to sit still, now imagine like 5 of them trying to get them together to sit still in the same orientation and all these different things. So it could be really, really hard to solve a complex with crystallography, although it can also be hard to solve it with cryo-EM. But with crystallography, it's a lot harder typically. So often what you see is that people actually solve structures of like the smaller parts of the parts in crystallography. So you get like high resolution structures of like these individual proteins, and then you can place them into like a lower resolution map from cryo-EM. Although lately now you can also get higher resolution data directly from the cryo-EM. But those low resolution structures can still be really helpful, kind of if you just get the broad outline. Then you have those higher resolution structures from the X-ray crystallography that you can then kind of place into that cryo-EM, that weaker blobby thing. So yeah, there's also a technique called NMR, which can be used for some really small things that are like really flexible and things like this. It's really good for, not so good for bigger things, but it kind of works like by the molecules kind of like tumbling and stuff, and so they have to be small enough to tumble, and then you need a lot of the sample. But yeah, it is used for some things. But yeah, the 2 main ones are really X-ray crystallography and cryo-EM. So remember, with crystallography, you're using X-rays. You get your protein to crystallize, which takes a lot of trial and error and trying out different things to try to make your protein crystallize. There's a lot of robots involved trying out different like cocktails, which is the liquid that you kind of like dissolve the protein in, and then you like let the water evaporate and stuff, or there's other methods too. But basically trying to promote this protein crystallizing, and then you have to find the right conditions, and then you have to optimize. So lots of screening and stuff. Then you send X-rays at them. The X-rays are going to interact with all of those different copies that are in the same orientation. So these signals are going to add up, or they're going to like cancel out. And the places they add up then lead to a stronger X-ray that gets detected by the detector, and then you have to work backwards from those detected the spots, from the detected rays, to figure out where they were coming from. And where they're coming from is going to be the electrons of those atoms, and so you get an electron density map, and then you build the model into the map. With cryo-EM, what you do is you take your sample, and all these proteins and everything is just like tumbling around and whatever they want to be doing. Then you flash freeze them really, really, really quickly on these grids. And these grids are basically, they have these little holes, and so you get these thin layers of this vitreous ice with the protein or whatever in it. And so this ice is going to be like, it's glassy. And so what's going to happen is your protein is going to be, like, as long as the ice isn't too thick or too thin, which is a big issue with the cryo-EM, then you're going to have your protein in all these different orientations. Potential problem is that sometimes there's like preferred orientations, where especially if, depending on the ice thickness, the particle might want to orient in specific ways and not every way. And therefore you can get like bias in which directions you see, and so you could end up with more images in like from one angle than you need, but less of another angle that you need, and so you can get like differences in the resolution in various directions and complicated things like that. The electrons are going to interact with the electrons and with the protons, so in the nucleus, and you're going to get the signal detected, and you will get be able to build from that. You're going to get your Coulomb potential map. And what you're going to also do is you're going to basically try to figure out, okay, well, which of these molecules which of these are actually particles, what's just crud, which of these look similar, can we then average the ones that look similar, and things like this. But you don't rely on the protein having to be in the same shape or in the same orientation. As long as there's enough of them in that orientation or enough of them in that shape, you can then average that information together. But you have to have enough of them in that shape or in that conformation. And so with Cryo-EM, you're going hole by hole by hole by hole by hole by hole in that grid, taking lots and lots of these images. And so there's like massive data sets that are generated, and lots of like time and power and stuff that's needed in order to do it. But hopefully then you get enough of these images, you're able to do all your reconstruction and stuff like this and get structure. So X-ray crystallography is still a thing, and it's still really, really great for doing smaller things, getting high resolution, especially if you know the conditions, like if the conditions for a protein to crystallize have been worked out, then you can just make modifications and get it to crystallize in the same conditions sometimes, and that's going to be easier. It's really good, like you can like soak a drug in there and like get the structure with the drug bound if you already know how to crystallize it, that sort of thing. But if you're trying to get it to crystallize for the first time, you might have to go through a lot of different screening as well as making different modifications to the protein, maybe like cut out a loopy bit in order to try to get it to stabilize more so that you can get it to stand still so that you can get it to crystallize. With Cryo-EM, there's also like optimization of the protein, of the sample prep, lots of optimization, lots of like biochemistry that goes into this sort of thing. It's not just like drop it on a grid and go. But yeah, so really cool stuff. And Cryo-EM, you can also, it's often used a lot too with like membrane complexes and things. You often do like nanodiscs where there's like little lipid around the protein. Cool stuff. But yeah, so hopefully that helped you kind of just understand the similarities, differences, the overview of these techniques. And if you want to explore them, they're cool.